Good morning, good afternoon, good evening, depending on the geographical locations. And thank you so much for being with us for this Global Virus Network series of webinars on the forefront of virology. And we are very grateful to Dr. Kanta Subbarao to be with us today. She is speaking from Australia. So first, these webinars are open up to, I would say, everybody. That means GVN members and non-GVN members. And so I just want to briefly introduce the Global Virus Network. So this is really a science-driven network which merged now 55 research centers worldwide and 10 affiliates in 33 countries. Again, this is really a science-driven network which aims to bring together the best experts in virology, working on all human viruses and obviously the connections with animal viruses with zoonosis. And so our activities are really focused on dissemination of knowledge with such webinars. We have annual meeting where we gather all the heads and scientists of the Global Virus Network to design, delineate research projects, cooperative research projects. We have been organizing task force on specific viruses, such as in the past on Zika, chikungunya, but also HTLV1, in particular, in fact, in Australia. And now, obviously, the Global Virus Network is very much focused on the COVID-19. We have activities on education, training, which are very important. Really, it's about shaping the next generation of virologists, be in basic research, but also obviously in medicine and in public health. We are very much focused also on advocacy with the GVN position papers on the most important topics. And I would encourage you to look at the GVN website. So this is the context of the Global Virus Network, and these webinars are very consistent with our views on, again, on the dissemination and sharing of knowledge. So in this context, it's a great pleasure to introduce Dr. Kanta Subbarao. Dr. Kanta Subbarao has a very interesting career track. She has been educated in India and then moved to the US. And she went through many institutions, actually, including St. Louis, Oklahoma, then Cleveland. Then she went also to McGill University in Quebec, then came back to Emory University and really acquired a lot of recognition. And then she moved to the NIH. She became in particular the chief of the emerging respiratory virus section in the laboratory of infectious disease at the NIAID. Dr. Kanta Subbarao is a highly recognized expert, and this has led us to be recruited in Melbourne as a professor in microbiology and immunology in the University of Melbourne, and to become since 2016, the director of the WHO Collaborating Center for Reference and Research on Influenza within the Peter Doherty Institute. And I should say that the Peter Doherty Institute with Sharon Lewin, with Damien Purcell, is one of the very active GVN centers on many topics. So Dr. Subbarao research has been focused on the new emerging viral disease, and in particular on influenza, on severe acute respiratory, on SARS, and on MERS. And again, she has acquired a lot of recognition on this. And this is shown by the number of publications, about 175, the contribution to many societies, and the editorial board of PLOS Pathogens and MBio. So really, it's a great pleasure to welcome Dr. Subbarao for this webinar, and we do look forward to listening to your lecture. Dr. Subbarao.
Thank you very much. I really appreciate the invitation to speak today. And I will now share my screen. Okay. All right, so hopefully you can see my screen now. And again, I thank you for the invitation to speak to you today. And before I begin, I would like to acknowledge the traditional owners of the land on which I live and work, and pay my respects to their elders past, present, and future. So I'm going to talk today about COVID-19 through a SARS-1 lens, because when I was at the NIH, I had a pretty active research program on SARS. And what we've seen over the course of the last several months is that this is obviously a very different scale of an outbreak, of pandemic, but there were some principles that were established through our work on SARS and MERS that apply to COVID-19. But before I get into the science, I just thought I would give you a brief update on what has been happening in Australia. And this is a epi curve starting from, let me see if I can, just a second, let me get my laser pointer. There we go. And this is an epi curve starting back in January, going to the 22nd of July, which was yesterday. And you could see that we had a peak of activity around late March. And things were actually going quite well for a while. And most recently, starting at the end of June, so roughly for the past month, we've had a gradual increase, and we have quite a significant number of cases now rivaling or exceeding the first round. And this is something that is probably happening in several other parts of the world. And here is a color coding by the states and territories in Australia. And you can see that almost all of the activity in this second wave or second round of cases has been from Victoria, which is where Melbourne is. This is a summary that is updated regularly at the Australian Government Department of Health website that packs in a lot of information about the total number of cases, deaths, and recovery, as well as the number of tests that are being done, the daily reported cases, and on a map of Australia indicating where the cases are. So that is just by way of an update of where we are, quite different from a month ago. And we're hoping that this current increase will actually be brought back under control with public health measures. So getting back to sort of our lessons learned from SARS-1, I'm going to start with the report from Wuhan from Professor Zhengli Shi's group. That was published in Nature, where they described a cluster of cases of atypical pneumonia. And this is by way of saying that in China and in many other countries that had sort of lived through SARS, people had established a surveillance system to identify clusters of atypical pneumonia. And in fact, that is how this first cluster of cases of people that were admitted to an intensive care unit, all but one of whom had been involved as market peddlers or delivery men at a wet market, who presented with respiratory symptoms, and the etiology of which was then explored. And what they reported was that they inoculated clinical material into vero cells, saw a cytopathic effect. They demonstrated coronavirus-like particles on thin sections by EM, and were able to sequence the amplicons using coronavirus primers. They compared the sequence with available sequence data. They showed that the receptor was identified as angiotensin converting enzyme 2, and showed serologic evidence of infection in recovered patients. Now, much of this was made possible by the surveillance program for atypical pneumonia that was established after SARS and avian influenza, and also the fact that they had a very active coronavirus, zoonotic coronavirus program. So the virus itself, this is just a schematic. As all of you know, there are only 4 structural proteins: the spike, the envelope, the membrane protein, and the nucleocapsid protein. But the genome is a very large RNA genome of 30 kb that encodes a number of non-structural and accessory proteins. So when I got into working on SARS was right after the CDC announced that it was that the causative agent was a coronavirus, and I was at the NIH, having just moved there from the CDC, and we offered to try to set up animal models. And so, of course, you know, I'm actually an influenza virologist by training, but had worked in a respiratory virus lab for many, many years. So we were quite used to inoculating mice intranasally with different pathogens, and then sacrificing the mice at serial time points and removing their lungs and nasal turbinates to see how much virus was present. And so these were young BALB-C mice, young adult BALB-C mice that we inoculated with a range of doses of virus. And we showed, in fact, that the virus replicated in both the upper and lower respiratory tract, especially at the higher doses. In that same experiment, one of the things that we looked for was whether the animals that and all the animals survived infection. So we looked to see whether they developed a neutralizing antibody response. And in fact, they did. So we'd set up a neutralizing antibody assay and were able to detect antibody in these mice. And we then took the survivors, which was essentially all of the mice from the first experiment, and we asked whether primary infection would protect them from subsequent challenge. So we brought in an extra set of naive mice and challenged the previously infected mice and the naive mice with 2 different doses of SARS coronavirus. And you can see that the animals that had been previously infected were protected from infection compared to those that had not previously been infected. We also took serum from mice that had survived their primary infection and administered that to naive mice. And these are the titers that were achieved in the naive mouse. And then we challenged those mice along with the group that got non-immune mouse serum and demonstrated that passive transfer of the immune serum to naive mice protected them from challenge virus replication in the lungs. Around the same time, my colleagues at the NIH, Ulla Buchholz and Peter Collins, did a very elegant experiment in which they took each of the 4 structural proteins of the SARS coronavirus, and they expressed them in a bovine human parainfluenza virus type 3 vector. They also tried combinations of S, M, and E, or M and E, and in empty vector control, and SARS coronavirus as a positive control. They vaccinated or infected hamsters with all of these constructs and demonstrated first that only the inocula that contained the spike protein of SARS, which was the intact virus, and the constructs, the vectors that express the spike protein, elicited neutralizing antibody. And they also challenged these hamsters with SARS coronavirus and demonstrated that only those animals that had seen a spike protein of SARS were protected from challenge, and this is compared to the empty vector control. So these 2 experiments established that mice that recovered from SARS coronavirus infection were protected from reinfection, and this now has been demonstrated with SARS coronavirus 2 infection in rhesus macaques. We also demonstrated that neutralizing antibody conferred protection, and this has also now been demonstrated with SARS coronavirus 2. And the experiment from Ula and Peter's group showed that the spike protein was really the only significant target of neutralization and as a protective antigen. We went on to develop a number of different animal models at the NIH, and this included young and older mice, and I'll talk a little bit about these. We tested a number of different strains of mice, including those with various knockout of the innate immune system that I will not discuss today. We developed a hamster model and also tested a number of non-human primate species. So the young mice, the young adult mice that we inoculated with SARS supported virus replication, and they recovered completely. They cleared the virus by about day 7. And there was very little pneumonitis. You could see some viral antigen in the lungs and viral RNA in the lungs, but it was really a very mild illness. And so there was a lot of sort of hand-wringing about the fact that there really wasn't a lot of clinical disease to follow. And so in the course of some of our experiments, we found that there was an age-dependent effect on the replication of the virus and on clinical signs. And so we went on to develop an older mouse model. We called them aged mice, but I'm told by the people that study aging that a 12-month-old mouse is not an aged mouse. It needs to be 18 to 24 months old to really call it aged. So in 12-month-old mice, when we inoculated them with SARS, and that's this lower graph here, you could see that there was a significant weight loss. We saw viral replication, of course, as before, but this time we could actually see antigen and alveolar damage going out much further. And that you can see in the slides below, indicating that there was much more multifocal interstitial inflammation in the lungs of these older mice. We went on to explore this in greater detail to look at the innate immune response as well as the adaptive immune response. And all of this is compiled on this one slide, showing in the mustard color the virus replication, virus titer, and the pneumonitis actually comes up after the peak viral replication is over. And we could see, we've studied the T cell response as well as the antibody and innate immune responses. One of the things we looked at was whether the pneumonitis whether the viral clearance would depend on CD4 or CD8 T cells. And a little bit to our surprise, actually, so this is in the 12 to 14-month-old BALB mice. We infected them with SARS or with a mock infection. And starting at day -3 and at day +3, 7, and 10, we administered anti-CD4, anti-CD8, or a combination of anti-CD4 and anti-CD8 antibodies and an isotype antibody. And sacrificed the mice at different time points, which are in blue is day 2, in brown is day 7, in green is day 9, and in purple is day 12. And you can see that the animals that had their CD4 T cells depleted actually cleared the virus later. There was delayed clearance in those animals. And it's very profoundly seen with the combined CD4 CD8 depletion, but also with just the CD4 depletion. The other strategy that we used to try to develop a more robust mouse model for study of antiviral drugs and vaccines was to mouse adapt the virus. And so we did this by intranasal inoculation of the original strain, which was the Urbani strain. We administered that intranasally to 3 mice, waited 2 to 3 days, harvested the lungs, made a 10% weight by volume homogenate, and administered that to the next set of 3 mice. And we did this iteratively until passage 15, at which point we had a virus that was lethal for mice at about 4 logs. So this is just indicating the weight loss, and these are these groups of mice died at day 4. When we looked at the virus replication, the black bars are the Urbani strain prior to adaptation, and the white bars are the adapted virus. And these are a lethal dose and a sublethal dose in gray. So really after day 4, we had no survivors following the lethal challenge dose. And the viral replication went up by almost 3 logs in the mouse-adapted virus. So we did find, though, that, and here you're looking at immunohistochemistry on day 1 and 2, and on the next slide 3 and 4 for SARS on the left, and the mouse-adapted 15 virus on the right. And you could see this is staining for viral antigen in pink. And we found a lot of viral antigen in the lungs of the mouse-adapted virus infected mice. And although it's not fair to look at, interpret in histology or histopathology from an IHC strain, you can, in the interest of time, I haven't included the straight path slides, but you can see that there was much less inflammation in the MA-15 mice. So what we found was that the virus was adapted for increased virulence and lethality in BALB-C mice, and this lethality was associated with viral replication to very high titers in the lungs, but was not associated with significant inflammation. We sequenced that virus and found that it involved 6 mutations in the viral genome, and a recombinant virus with the 6 mutations recapitulated the lethal phenotype seen with MA-15. And we did this in collaboration with Ralph Barrick's lab at UNC, and he made a recombinant version of this. So this virus could be used to evaluate the efficacy of vaccines in mice, and in fact, Ralph's lab has continued to use the MA-15 virus in the older mice, so that they combine 2 of these models. So moving on to golden Syrian hamsters, we infected hamsters with a much lower dose of virus. Here we inoculated them with 3 logs of virus intranasally. And this is looking at the viral titers in the upper and lower respiratory tract. And this is virus titers in log to the base 10 TCID50 per gram over a series of time points. And you can see that we saw robust viral replication up to 6 logs or 8 logs, 6 to 7 logs in the lungs of hamsters through 7 days of post-infection. And on the right-hand side, you can see that these animals developed very robust neutralizing antibody responses. And in looking at pathology here on the right and immunohistochemistry in the lungs and nasal turbinates, you can see how much viral antigen, which is in pink, is demonstrated in the respiratory tract. So this the hamsters ended up being our most favored model for studying the efficacy of vaccines. We also looked at a number of different non-human primate models, and in one study we compared rhesus, homologous, and African green monkeys. We gave them 6 logs of virus intranasally and intratracheally, 1 ml in each site, and we collected samples from the upper and lower respiratory tract daily from the upper and every other day from the lower respiratory tract as tracheal lavages. And you could see that in our hands, the African green monkeys were actually the most robust of the 3 models, but none of them developed clinical signs of disease. So the corresponding observations with SARS coronavirus 2 actually are very similar to our findings with SARS-1, and that is that the hamster model shows robust infection of the upper and lower respiratory tract. This is work from a couple of different labs, the lovely paper from Jasper Chan in CID. Non-human primates can be infected, but the findings vary amongst rhesus, cynos, and AGMs in our hands, and certainly with SARS coronavirus 2, non-human primates can be infected. Mouse-adapted virus and age dependence is something that is being explored now by a couple of different labs. So we use these animal models to study a number of different vaccine approaches. And in my lab, we tested about 9 different vaccines and about 4 different antibodies over the years. And now there are obviously a number of similar approaches for SARS coronavirus 2 vaccine approaches. And this is from the coronavirus vaccine tracker in the New York Times, indicating how many vaccines are now in preclinical development for SARS coronavirus 2, including how rapidly in the course of just a few months, so many vaccines have gone into phase 1, 2, and into clinical trials. So the good news on the vaccine development front is that the optimal target for vaccines was known to be spike or S protein. The new vaccine platforms are available, including messenger RNA, stabilized proteins, and vectored vaccines that can be adapted rapidly. And what's different from back in 2003 and 4 when we were working on SARS is, of course, that the global funding consortium that was preparing for virus X, and that is CEPI. And here we are with virus X being SARS coronavirus 2. There's also lessons from Ebola with the new willingness to evaluate a vaccine in the midst of an epidemic. The challenges are that there are no licensed coronavirus vaccines yet, so there's no clinical experience with safety, immunogenicity, or efficacy. Only 2 of the SARS vaccines went into phase 1 clinical trials, one in China and one at the VRC at the NIH. Both of them were in small groups of people. There were some specific safety concerns that were raised in monkey and mouse studies with the SARS vaccines. And the longevity of the immune response is a matter of a lot of discussion. And of course, the $1 million question is whether SARS-2 will become a seasonal respiratory virus and whether we will need to have an ongoing vaccine program. So one bit of data that we had on longevity of the antibody response is on the next few slides. And this was a study that we did in hamsters, where we infected hamsters with SARS, or we vaccinated them with one of 2 different vaccines. One was a modified vaccinia virus expressing the spike protein, and the other was a purified spike protein. So a group of 6 hamsters were given these 3 immunogens, and they were followed for 8 months. And what you're looking at is the neutralizing antibody response over the course of this period of 8 months. And you can see that the tiny purple dots, which is the last group, are the ones that were infected with the wild type virus. And you could see that the antibody response was fairly well sustained, as was the response to the purified spike protein. The MVA expressing spike, the spike was the least immunogenic of the lot and actually dwindled down to the lower limit of detection, which is this dotted line. We also challenged these animals at 2 different time points, one at 1 month post-immunization, where you can see that animals that had been previously infected with SARS, got the spike protein or the MVA expressing spike, all had a very good protection from challenge compared to the 2 negative controls, which were PBS inoculated hamsters or MVA, a blank MVA infected hamsters. And these were lung titers 2 days after challenge. This was given where challenge was administered 1 month out. The other group of hamsters were challenged 8 months post-immunization. And you recall I showed you what the neutralizing antibody titers were. And what you have in darker colors in each case are day 2 post-challenge. And in the lighter color is day 14 post-challenge. And this was to see whether the 8 months later, if you challenged animals, would you have viral clearance, and also whether there'd be any long-standing viral replication, or enhanced disease. And so what we see here is that animals that had had a primary infection were well protected, as were those that got the spike protein. And in contrast, the MVA spike and the PBS controls did not, but none of the animals had sustained replication 14 days out. This is a different study that we did in collaboration with GSK using a whole inactivated vaccine given without or with an AS01B adjuvant, and a control here is a BPL inactivated influenza vaccine with AS01B as an adjuvant. The positive control here is SARS coronavirus infection. So in all of these, we challenged animals 4 weeks after dose 2 or 18 weeks after dose 2, and harvested the tissues 2 days post and challenge. And you can see that if you focus first on the white bars, which is the early challenge, 4 weeks after immunization, the vaccines work very well with and without adjuvant at a low dose or a higher dose of spike. However, when the animals were challenged 18 weeks post-vaccination, only the animals that got the adjuvanted vaccine were well protected. We did not see any evidence of pneumonitis or enhanced inflammation in the lungs of animals. These were lung tissue looking 5 days post-challenge, and this is following the late challenge, 18 weeks out. At the bottom here are the previous SARS infection. On the left is histopathology, and on the right is immunohistochemistry, so we didn't see any evidence of viral antigen or inflammation. And nor did we see that in the animals that had been immunized with the spike protein with or without adjuvant. In contrast, in animals that got the BPL inactivated influenza vaccine, you see a lot of inflammation and antigen. So you fast forward now to 2020, where I've relocated and now I'm in Melbourne. And here we're working on neutralizing antibody assays, antiviral drugs, and 2 clinical studies in healthcare workers. One to look at the risk of infection in healthcare workers that are occupationally exposed to SARS-CoV-2, and the use of BCG vaccine to induce trained immunity. So just to give you a snapshot of the neutralizing antibody responses that we're detecting, and this is done with a microneutralization assay, very similar to what we developed for SARS and MERS. And you could see that there's clearly by 14 days post-infection, we're detecting a very robust neutralizing antibody response. At the Doherty Institute, among many other projects, and I'm only talking about those that I am directly involved in, but there's a whole host of work going on at the Doherty. And what we're working on is an antiviral program where where compounds are submitted for consideration. We have a scientific review panel that reviews and scores these for the drugs that are submitted for consideration based on a number of different criteria. And if they meet our criteria of availability, expected efficacy from other data, practicality, and cost effectiveness, those that are scored well go on into a screening assay in Vero cells across a range of concentrations against 100 TCID50 of virus. And if we see inhibition of viral infection, we would then confirm that and do cytotoxicity assays as well. Those that show promise could then go on to an in vivo testing in a mouse or other model. And we also have the ability in collaboration with many others at the Doherty Institute to evaluate the mechanism of action of these drugs. So should we have, this is my assessment of where we are in 2020. And one question that we can ask ourselves is, should we have seen this coming? So certainly pandemic preparedness activities were largely focused on influenza, although the zoonotic risk of coronaviruses and other virus families like Nipah and Ebola was known. We've had no licensed coronavirus vaccines or antiviral drugs, despite a lot of work that went into SARS and MERS. What was different this time is the efficient transmission by the respiratory route and spread from asymptomatic individuals, neither of which was a feature of SARS or MERS. What's worked well is the capability to identify the virus and to test for it. We've had many lessons from SARS and MERS, as I've said. There's been a rapid sharing of scientific information, and the GVN network is one such example. And certainly in countries, influenza pandemic preparedness plans were adapted. Certainly social distancing was not a big part of the influenza preparedness plans, but will certainly be of note in the future. And government decisions in many countries were well informed by science. What should we keep in mind for the future? That there are other coronaviruses, influenza viruses, and paramyxoviruses with pandemic potential in animal hosts. And it just reiterates the importance of One Health, the fact that human and animal public health and the environment are closely intertwined, and we cannot achieve one without paying attention to the others. So with that, I will close and acknowledge the people in the top part of the slide are the people that I worked with extensively over the years. I had a phenomenal group of colleagues and collaborators at the Laboratory of Infectious Diseases in NIAID. Brian Murphy was the lab chief at the time. We collaborated closely with the infectious disease pathologists at the CDC. I've had a long-standing, very good collaboration with Ralph Barrick. We tested vaccines from many different developers in academia and industry, as well as monoclonal antibodies that I haven't talked about today. My lab here, these are the members of my lab here, and they are working tirelessly on a number of different projects. I've got many wonderful colleagues at the Doherty Institute that I would call out. We've had funding support from the NHMRC in Australia, from two philanthropy foundations, and the clinical studies that we're involved in are funded by the SEERS network of NIAID. And I work at the WHO Collaborating Center for Influenza that's supported by the Australian Government Department of Health. So with that, I will stop and stop sharing my screen. And I think, and hand it back to Christian.
Well, thank you so much. Thank you so much, Kanta. This has been a great lecture. And I believe that really you have very nicely illustrated how we need to, to really retrospectively analyze the data and to really use this for the prospective assessment of this ongoing pandemic. So thank you so much. So we will have questions. So maybe you can start looking at this, Kanta.
Okay, so I see a question about which vaccine platform gave the most promising results in our lab, and which animal model was most useful for preclinical trials. So I would say we actually, when we were approached to look at different vaccines, one of the things that I asked each of the people that asked us to look at their vaccines to give us the ability to compare vaccines head to head. And unfortunately, most people didn't, and of course we didn't have the time to do so. So I would say that all of the vaccine strategies that we looked at did induce neutralizing antibody, and those that induced neutralizing antibody, or at least the ones that induced robust neutralizing antibody, were protective. We found that inactivated vaccines and spike protein vaccines that were adjuvanted provided very robust immunogenicity and protection. Probably the one that was not as robust in our hands was an MVA expressing spike, but all the others looked really good. The second question was which animal model we preferred, and I think I said this before, that we preferred the hamster model because there was very robust replication and immunity. The next question is, for your vaccine studies, have we considered boost immunization, and have we determined memory B cell responses? So we certainly did in almost all of the vaccine strategies that we used, except a live vaccine, which I didn't talk about. We had a Delta E live vaccine that was made by Luis and Juanes from Spain. All the others were done in more than one, with booster doses. And we didn't look at memory B cell responses. We didn't have the technology to do it with the hamsters. And we saw such robust antibody responses that we limited ourselves to that part. Do I believe that one antiviral drug can protect from virus? So that's a good question. I haven't done any studies with antiviral drugs in animal models. I didn't do them before with SARS and haven't done them yet with SARS coronavirus 2. But I would say that it's getting complicated. As long as we are repurposing drugs, and because remdesivir is now very widely used as a standard of care, I think we are going to have to look at combinations. And certainly there's great evidence from hepatitis C and HIV and other areas that more than one drug is often better than a single drug. So I think we will be looking at combinations. Ramesh, great. Any thoughts on possible ADE? That's a good question. And I think we have to be concerned about the possibility of ADE. I must say that we didn't see it in mice or in hamsters in all the studies that I did with SARS, but others have certainly reported finding this. And I would point out that the vaccines that were tested showed actually protection from viral replication, but what was found was evidence of inflammation in the lungs. So that remains a possibility. And I think, you know, and I'm part of a group that wrote a summary on this that should be published fairly soon. So I think I would watch for it, and I would look to see if there's a balanced immune response to a vaccine. I think that would be one way to protect against the possibility of ADE. Of course, the prior experience with inactivated RSV vaccine is also in the 60s, is part of the reason we always worry about any new vaccine. So I would say that that in theory, the risk exists. I haven't seen any evidence of it. And one of the reasons that I was highlighting the longevity of the antibody response and challenge at a late time point was that if you were going to see that, it would be after the decay of antibody. So I think that that's something that we have to just watch that space. So I think I answered the ADE one, yep. So did we use the mouse? No, we did not use the mouse-adapted SARS-2 in hamsters. We just used the Urbani strain, the unmodified Urbani strain. And so what's interesting with hamsters, I was told that it wasn't worth weighing hamsters because they have cheek pouches and they hide variable amounts of food in their cheek pouches. And so we never actually weighed them. But I see from people that are working with SARS-2 that they're seeing weight loss with SARS-2. With the high community transmission, could people who are already infected without their knowledge have produced potential neutralizing antibody? If so, do they need to be vaccinated when the vaccine is ready? So that's a good question. When we first started looking, when we developed the neutralizing antibody assay and we tested sera from people that were admitted with SARS-2, I was actually a bit surprised to see that in some people we were detecting neutralizing antibody in the first blood draw, which was, you know, day 7 or 8 post-infection. So I think there's a neutralizing antibody. Of course, the incubation period for SARS is much longer than it is for flu. It's an average of 5.6 days. And so some people have actually been, but if we count their illness from the time of onset of symptoms, they've probably had 10 to 12 days of infection. So having said that, there are times that we detect antibody in people that were infected without their knowledge. But I think there's some evidence that is published now from other groups that suggests that the antibody response in people with very mild or asymptomatic infection may not be very long-lived. So I do think that it would be very interesting to vaccinate people that have that history to see if, in fact, they get a more of a recall response when vaccinated. What do I think is behind the second wave in Victoria? So I think it's complicated. I think from what I understand, a lot of this has come from some breaches in the quarantine. So what had happened was that when people came to Australia, travelers came into Australia, they were asked to quarantine. And initially, they were asked to quarantine at home. And eventually the government actually changed that to a hotel quarantine. So the second wave in Victoria seems to be linked to some breaches in the hotel quarantine that were unfortunately missed before they got into the community. So we now have a number of there's a number of clusters of community transmission, many of which have been actually linked to the quarantine spill. But in addition, there have been outbreaks in meatpacking plants, which has also been seen in other countries, including Germany and the US. So there is now some community spread. In previous SARS-1 work, were different innate immune deficient mice susceptible without viral adaptation? Yes, so there's a paper that originally that Will Glass from Phil Murphy's group did in our lab in collaboration with us, looking at a number of different, we looked at RAG knockouts and a whole series of different knockout mice. In our hands, actually STAT1 knockout mice got really quite sick and had very persistent replication. And we had a publication with Matt Freeman and Ralph Barrick that explored the basis for that. And we looked at interferon lambda as well in those mice. So that work is published.
Thank you very much. I believe that you have covered the questions. Now, I just want, before closing, to make just 2 comments, from what you have presented and from the questions and your answers. To be honest, I'm very unclear regarding the ADE, because you very nicely illustrate that we just don't know exactly to which extent this might be a barrier to the effort for vaccinations. And yet we have all these announcements on phase 3 vaccinations, which are being initiated. And I have some difficulties to understand how we can move so fast for this phase 3 with these still pending questions, but that's a personal comment. The second is that you showed a very interesting slide at the end on your assessment of what has been wrong, I would say, and what works. And I found it very optimistic when you said that governments have followed scientific advice. Well,
some governments.
Some governments, I would say a handful of them. And we have good examples of large countries where this is not the case. Now, the reason why I bring this up to the conclusion is that we are really working with the GVN on a kind of a forefront vision paper with all the colleagues to delineate how we should be maybe better prepared, including for potential so-called second wave in the future. So I found this comment very, maybe you can help us on this. So thank you so much. It was again a great presentation. Thank you for the attendees and for the questions. And obviously this series of webinars will go on with many exciting lectures which are being planned. And thank you to all of you. Have a very good day, afternoon, evening, and be safe, as we used to tell, and we keep in touch. Thank you so much. Bye.
Thank you very much.
Thank you very much.
Thanks.